Hello, AP Biology students. Let's go over the third section in Unit 4. And in this section, we're going to review what the cell cycle actually is. The cell cycle is a set of stages that takes place when a cell divides. And a lot of times we show this in a diagram that looks like this. And these diagrams are usually in the shape of a circle since these cells will continuously divide. The cell cycle is going to be divided into two main parts, interphase and M phase. Interphase is typically when the cell goes through normal functions, and M phase is when the cell is actively dividing. Our first major portion of the cell cycle is interphase. You can see interphase is going to be shown here in this purple color. A majority of a cell's life is going to be spent in interphase. This is typically when the cell just goes through normal daily functions and just exists. Now, there are a couple different parts within interphase that are specific to how the cell will divide in the future. These three main parts are called G1, S, and G2. In G1 phase, the cell is going to recover from its previous division. This is typically the phase where the cell is going to also grow in size a little bit, but it's basically going to increase the number of organelles just as a recovery method so it can survive because it just got done dividing. If you can see here, G1 phase follows M phase, which is mitosis. So this is when the cell is just divided and the cell is just going to go through normal functions. It's important to remember when the chromosomes condense in the cell cycle. The chromosomes will not condense during interphase. So during G1, S, and G2, again, the chromosomes are not condensed. I have to throw this one in here. Chromosomes normally, chromosomes during mitosis. Perfect. S phase stands for the synthesis phase, and this is when replication occurs. Replication is when the DNA copies itself, making an identical copy. Before S phase, the chromosomes only have one chromatid. After S phase, the chromosomes will have two chromatids, and these two chromatids will be attached at their centromere, and they will be sister chromatids. Again, the chromosomes are not condensed yet. During G2 phase, the cell is going to synthesize proteins it's going to need for cell division. Basically, this is a prep for the next step, which is the cell dividing. And once again, the chromosomes are not condensed yet. When it says chromosomes are not condensed, what it means is the chromosomes haven't condensed yet in the X-like pattern that you might recognize them in. You can see here in this cell, we have chromosomes or DNA that's not condensed. It almost looks like a bowl of spaghetti. So when we say the chromosomes aren't condensed, they're unraveled and they're able to be read, which makes sense because the cell is going through normal functions. It needs to read the DNA. It will condense the DNA when it's ready to go through mitosis. Now, there is one minor step within G1 phase that I didn't talk about. It's called G0 or G0 phase. This is also called the lazy phase. This is where a cell will not go through mitosis and will continue doing normal functions and surviving, but it just doesn't divide. There are mechanisms and proteins to stop it within the G1 phase so it doesn't divide. I like to explain the importance of this phase as being it's trading its ability to divide to have a very specialized function. This is golden. Cells in G0 phase, yeah, they're just not dividing.